how did we get to this point in personal genomics? Perhaps the better question really is, what is the future of genomic medicine? There's no better person to educate and inform us of all of these questions, the guy who really has a whole lot of answers. My friend, Dr. Neil Lamb, Vice President of Educational Outreach for HudsonAlpha. Neil?
Good evening. Thank you, Liz. It's a real privilege to be here with you and to set the table for you, to give you a little bit of background about how we got to this place where we're now talking about personal genomics. And the story that I want us, the analogy I want us to think about is that of a puzzle. So I don't know how many of you are puzzle people, but when I start with a set of puzzle pieces, I generally try to identify something that I can easily put together and then build around the outside and then save the inside for last. The inside always seems to be the hardest part, and I have to spend the most time finding the fit for the inside. That's an analogy that I want you to hold in the back of your mind as we talk about this process of genetics and genomics. What is it that has gotten us to this place where we now stand, where we're talking about impacting healthcare and agriculture and energy and all sorts of other fields? So I'm going to take us all the way back to 2000. So while many of us were frantically preparing for Y2K, scientists around the world were working on sequencing the human genome. So a genome, just as a reminder, is the collection of all the DNA that's found inside an organism's cell. So toads have genomes, cows have genomes, people have genomes. And the aim of the Human Genome Project was to identify all the genes inside our human DNA, and to identify where they're located, to interpret or read that code of life, that chemical code. There were several different people working on this from all around the world. And at that time, in the year 2000, if you were able to sequence a human genome, we couldn't have done it at that point, it took multiple years, but if you could have done it in the year 2000 with the cost of sequencing, it would have cost $100 million to sequence one individual's genome. Now, I want to introduce you to a group of scientists from Stanford University who were part of the Human Genome Project. They're actually seated here because we now have the privilege of having them as members of HudsonAlpha. So Dr. Rick Myers, who's here at table 13, Dr. Jane Grimwood and Jeremy Schmutz, who I think are at table 32, right over here. I can't see their table number. And Dr. Devin Absher, who I think is at table 17. This group was involved in sequencing about 10% of the human genome, working with the most advanced technologies of the time, giving us those first set of puzzle pieces, beginning to build that outer side. The Human Genome Project showed us that we could do big collaborative data projects, and that it actually created more questions than answers. So it was followed by other projects like HapMap and ENCODE, which really sought to say, okay, now we know how many genes there are. What do changes in those genes mean? Should we be worried if I find a change in someone's DNA? Is it part of the beauty of natural variation across the diversity of humans? Or is there something I need to pay attention to? And ENCODE said, what's the functional significance of that? So these big scale projects that carried on, in fact, iterations of ENCODE are still going on today. That brings us to 2005, when Lonnie McMillan and Jim Hudson incorporated HudsonAlpha. So we are celebrating our 10-year incorporation anniversary with this vision of bringing scientists and companies and educators together to explore, solve, and inspire. Groundbreaking was held in the winter of 2006. Some of you in this room are in that photo. And about 2 years later, we opened the building on a wonderful spring day 7 years ago this week. I would imagine many of you were at the gala that night, and what an incredible day and series of days that was. Now, if you've been following along, you've noticed that the number of gold coins showing the cost of sequencing a genome has been dramatically shrinking as we followed this. And so, in fact, by the time we opened the building, it was just a little over $1 million compared to that $100 million to sequence a human genome. And this was due in large part to advanced technology, so-called next-generation technology, that did the sequencing on a much, much smaller scale in parallel. There were multiple reactions taking place at once and drove costs down. When we opened the building, we had 2 of those cutting-edge machines here on site. And from that moment on, HudsonAlpha has always been committed to continuing to follow the new technology, to pursue the benefit of being an early adopter of the new technology. Late 2007 through 2010 was where we got our legs under us and we built our faculty. Dr. Jin Han, who is overseas tonight, came on board in late 2007. In 2008, we brought the team from Stanford, Rick, Jeremy, Jane, and Devin. And that was followed in 2009 by Dr. Greg Barsh, who is, I think, at table 31 or maybe, yeah, table 31. He came from Stanford. And Dr. Sean Levy, who's over here at table 6, who we recruited from Vanderbilt. And then in 2010, in the fall, late summer, we brought Drs. Greg and Sarah Cooper from the University of Washington. And they're right here, I think, at table 18. And that brought us to the number of faculty that we have had that have served us so incredibly well over the last 5 years. Taking genomics, the ability to rapidly look at large amounts of genetic information and apply that to an understanding of disease, agriculture, human health, and beyond. Now, at the same time, in Milwaukee, there was a young man named Nicholas Volker. You've already heard his name mentioned. You've already heard a little bit about him. We're going to watch a brief video. This is a clip from the Today Show in 2010 that highlights the challenge that this young boy faced and the inspiration and the willingness of a team of scientists and clinicians to stand in the gap to try to give him an answer. So let's watch that video.
To look at 6-year-old Nick Volker, you might never guess he had been close to death hundreds of times, and the struggle to save him will be a milestone in medical history. His mother, Amy Lynn Santiago Volker, says he is always upbeat.
He loves his mom and his dad with all his heart. He's such a loving little boy.
Nick was 17 months old when the first sign of trouble struck, a wound that would not heal. Over time, Mom realized he faced a life-threatening condition.
6 to 8 months after his first hospitalization, at that point he was down to 17 pounds. And he was always having pain.
At first, doctors thought Nick suffered Crohn's disease, an autoimmune disorder of the intestine. Surgeons removed his colon, but Nick continued to deteriorate.
Watch out!
Last March, Dr. Alan Mayer, a pediatric intestinal specialist at Children's Hospital of Wisconsin, took charge of the team handling the case, but they had little success.
We were never able to get his inflammation under control.
Dr. Mayer and others consulted with experts around the world. Everyone was stumped and thought Nick would die without a diagnosis.
The doctors were so perplexed by the case that they made an unusual and desperate request. Could the scientists here at the Medical College of Wisconsin sequence the some 30,000 genes in the boy's body to find out if one was mutated?
I was shocked. It's a big leap from what we were currently doing.
Howard Jacobs and the medical genetics team had only done basic research. They never looked for a disease in an individual. Mom was certainly on board.
I thought, let's do it. We need to find a diagnosis.
It came from being a shocking request to, we need to do this.
They found that Nick has a defect in a gene called XIAP, a condition known to affect only a handful of others in the world. The treatment was a cord blood transplant to give him a new immune system. There were many frightening complications, but Nick pulled through.
You know, he's the comeback kid, because I don't know how many times, over 100 times we've been told that Nick, won't make it to the next hour, the next day, the next month, the next year.
And because gene sequencing is becoming ever cheaper and easier, the doctors think Nick's case is a crucial beginning of a new era of medicine. For today, Robert Bazell, NBC News, Milwaukee.
A crucial beginning of a new era of medicine. You heard them say that as they sequenced through Nick's genome and looked at all of his genes, they were specifically comparing his genes to a reference genome, looking for changes. they identified a change in a gene called XIAP, and this gene encodes a protein also called XIAP. Now, in true HudsonAlpha education fashion, we actually have a hands-on for you to work on. On your table is this model. So I'm going to ask whoever's closest that you get it, and that you take a look at it and that you pass it around the table. This is a 3D, this is made on a 3D printer. This is a model of the protein XIAP. Proteins are made of amino acids, and the specific order of amino acids determines the shape of the protein. So I want you to pay careful attention to two parts of this protein. In the upper corner, there's a really small, it's actually magnetic, it comes off, a really small amino acid. It's one black sphere and one yellow sphere. This represents the amino acid cysteine. It's very small. It fits right in an important corner where the protein makes a bend. The shape of the protein, the structure of the protein determines its function. I'm willing to bet it goes right back in the upper where the number 203 is, for those of you that have taken it off and need to figure out where it goes. Those of us in this room have this amino acid. Now at the bottom, there's a larger amino acid that's docked to the base. This is tyrosine. Tyrosine is a much larger structure. Nick Volker has tyrosine in place of the cysteine at this position. So you can imagine if you try to take the cysteine off, you try to put the tyrosine in place, it doesn't fit very well. The protein has to change its shape to accommodate this amino acid. And when it changes its shape, it changes or loses its function. So it's this change in Nick's DNA that changes the structure of XIAP protein that led to Nick's disease. Now, those of you that are table sponsors, this is your take-home thank you from us. You may have to wrest it away from the hands of someone else at your table, but we want it to be a reminder of putting more puzzle pieces in the puzzle, of getting more answers, and of changing lives. You heard them say that Nick had a cord blood transplant that's similar to a bone marrow transplant that gave him a new immune system. And here's a picture of Nick in 2012. Nick still bears the very real scars and complications of all the surgeries and of the challenge of a transplant, but he is by and large a happy kid who can enjoy life in ways he never could without this sequencing. This is the power of reading our genomic information. Now, let me bring us back to HudsonAlpha and finish our timeline. You'll notice that the cost of sequencing at the bottom is now not even a full gold coin, it's a piece of a gold coin. And today, best estimates are that we're somewhere in the $1,500 range to do the sequencing. Dr. Levy can give much more specifics than that. That doesn't include the cost to interpret it. And that's where much of the cost today now lies. So that $1,800, $1,400 just gives you a list of A's, T's, C's, and G's, not the interpretation of that. As the Institute has grown, we've had 6 other spring benefits. And we've been able to talk to you about how our scientists are using the research to try to solve issues in cancer, neurodegenerative disease, psychiatric disorders, type 1 and type 2 diabetes, undiagnosed childhood diseases, which is our own first steps in clinical sequencing, and last year the immunogenomics program. At the same time, the scientists at HudsonAlpha have been hard at work understanding how changes in DNA alter their structure, their function, how they relate to health and disease, dealing with issues like discovering new genes for ALS, identifying biomarkers for kidney cancer, working on projects that help build the encyclopedia of what happens in our DNA. Our Genome Sequencing Center has been sequencing some of the world's most important agricultural crops and making a difference in the security and sustainability of our food supply. And our scientists also are answering basic questions about how life on Earth evolves and responds to changing conditions, and how things that seem as basic to our understanding of patterning, coat color, and coloration in animals links to broader issues of organismal development and health. Again, putting the pieces together. And in each one of these cases, more pieces gives us more insight into the application of the science. The science drives our understanding, and then whether it's agriculture or energy or healthcare comes along and applies that into the clinic, the farm, or the power plant. I don't know what lies ahead. I can't wait to see what the next several spring benefits look like in the stories that we tell.